All right, welcome everyone. Thank you so much for coming. I wanted to welcome you to the Naturalist Training on Aquatic Invertebrates. This is our 7th online training, including serpentine soil and associated grasslands. And we've also hosted one on nature interpretation, and we're so glad you're here. You can find our past recordings on the Keep Coyote Creek Beautiful YouTube channel. In this presentation, we will hear from Shelley Pneh about macroinvertebrates in our creeks, those little wiggly critters, many of whom can help us determine creek health. Hi, my name is Deb Kramer. I'm the Executive Director and founder of Keep Coyote Creek Beautiful, and I'll be your moderator tonight. I relish being out in nature, and it is nurturing and healing. Since I was a kid, I played in my backyard where I had creeks. And over the past 9 years, I've had the great fortune to build a community around Coyote Creek through Keep Coyote Creek Beautiful's events. One of my favorite activities is our BioBlitz event, where we explore a locale for a couple of hours and learn from naturalists about what we find. And Shelley is one of them. She's been out with us on a number of BioBlitz events, and we're so fortunate to have her here today to share her wonderful world of aquatic insects. Her talk will focus on California's water-based organisms that not only function as food, but also characterize the health of our streams. Shelley Pneh will share her excitement about these small but mighty creatures so you can apply your newfound knowledge to help determine the health of our waterways. So let me introduce you to Keep Coyote Creek Beautiful. We are a community-based organization, and we see Coyote Creek as a resilient ecosystem for all things to enjoy. And we do that by engaging, educating, and encouraging people to bring communities together to take action for, learn, and play along a healthy Coyote Creek. So what does that look like? Well, we host creek cleanups. We have BioBlitz events for people to perform community service and learn about nature. We even have had backpacking and nature walks. We also have a robust classroom program and work with college students and their classes. So now to introducing Shelley. So Shelley Pneh is a Bay Area native who pursued molecular environmental biology in college and ended up seeking outdoor environmental careers. She's currently an ecologist with Grassroots Ecology, love Grassroots, and focused on habitat restoration and environmental education. With a green stormwater infrastructure and pollinator gardens in Palo Alto. She also works on watershed wildlife corridor restoration of lower San Francisco Creek and upland marsh transition zones restoration at Shoreline at Mountain View. So definitely very much into working with young people and making things happen around restoration. One of the cool things about Shelley, aside from the fact that she served on the Keep Coyote Creek Beautiful board for a few years, is that she mentors the next generation of young professionals in the environmental field through AmeriCorps and Stanford master students, UC Berkeley grads, and local high school students. Concurrently, she is an aquatic entomologist, field tech with the Power Lab at UC Berkeley, with a focus on freshwater food web research, specifically in Northern California watersheds. And I'll let her talk more about that. And if you haven't seen any of her photos, she has contributed over 250 identified and photographed aquatic insects to CalPhotos, a digital natural history museum database. And I'll definitely be sure that we have a link for that in the notes from this YouTube presentation. So with that, I'm going to go ahead and turn it over to Shelley.
Thank you, Deb, for that kind introduction. I'll let everyone sit for a little bit as I try to share my screen. Thank you. Well, thank you all for joining us. This presentation is going to be about aquatic macroinvertebrates, a little bit about the ecology of what they do, I'll spend also a little bit of time talking about some special groups that you might find in the stream. And then my last part of my presentation is going to be discussing more about how we can add more aquatic macroinvertebrate observations to iNaturalist. So what is an aquatic macroinvertebrate? It actually encompasses a whole lot of different organisms that are aquatic. So the word aquatic means that it has to live in water for some of its life. Macro meaning that you can see with the naked eye, and invertebrate are all organisms without a backbone. So our most common invertebrate that we probably know of are insects. And this is kind of like a tree, an evolutionary tree of all of the insects that we know of in the world. And aquatic macroinvertebrates actually encompass a lot of these insects. We have pretty much over 50%. And also, not only are there other invertebrates that are non-insects that are found in the creek, we also can find other things that are not insects as well. So I want to kind of zoom out and bring you to a creek or stream ecosystem. So this is a very simplistic diagram of a stream, but it's basically broken into different areas. First, we have the headwaters, or basically where the creek originates when the water is coming out of the ground. They're usually found in very high mountainous areas. That's usually very covered. It's usually in the forest. So there's a lot of trees that are covering this small little creek. And then that means that we also have a lot of leaf matter that is falling into this creek ecosystem as well. And since this part of the creek is so shaded, most of the water is very cold. And then if you picture yourself as a molecule of water floating downstream, you'll sort of end up where more of these smaller streams connect together to form a larger stream. So this is what we call the mid-reaches. There are more and more sunlight as the creek gets wider and wider. More sunlight is hitting the bottom of the channel of the creek, which makes it so that there can be photosynthesis from the algae, the cyanobacteria that basically use the power of the sun to make sugars in their cells, and to which other animals can then eat this. Periphyton is a word that just encompasses like all of the microbiological stuff in the creek. So you have bacteria, fungi, algae, other organic debris. And so that is all of that. And then as you keep floating down the stream, it gets wider and wider. We kind of start to see that the shores are a lot more bare. You might see like a lot of sand and more mud. We don't really have many trees anymore, but we have a very wide channel with lots of water, deep pools, and we have a lot of fine particulate matter that's floating down the stream. So if you're thinking about like downstream Mississippi River, the water is actually very murky, and that's because like all of this fine sediment is flowing down into this larger body. Oops. Let me go back. And then with all of these different habitats along the water channel, you can see different assemblages of macroinvertebrates that are present in certain areas. And ecologists like to group these macroinvertebrates into different functional feeding groups. Basically, this is an easy way how ecologists can track how organic matter gets cycled through this ecosystem, through this food web. And you can have many different species that can be part of a certain functional feeding group. If you look to the diagram on the right, we can start at the very top where we were talking about the headwaters of the stream. Remember how I mentioned on how it's a very, very dim area, not enough sunlight's getting through, but there's a lot of falling leaves. And so you will find macroinvertebrates who are specialized on chewing up all of these organic matter, all of these fallen leaves, and making them into smaller and smaller particles. They will eat some of it, but they're also very messy eaters. So they will also leave behind a lot of breadcrumbs, if you will. And then there are other macroinvertebrates that actually eat these breadcrumbs. And so they will be following close behind these organisms and eating whatever they are breaking up. And then, of course, we always will have predators that will be eating other macroinvertebrates as well. So when you get to more of the middle reaches and where you get more photosynthetic activity and more algae formation, you start to find other macroinvertebrates that are more specialized to eating algae. We call those grazers. And grazers are kind of, just think about them as like your cows or your antelope, some big organism that likes to just mow down lots and lots of vegetation. So grazers are the macroinvertebrates of the creek's ecosystems that are doing the same thing. And so you'll find a lot more grazers in these reaches. You'll still find a lot of collectors who are the ones that are eating all the breadcrumbs, but you find less and less shredders, which were the macroinvertebrates that are the ones that are breaking up all the leaf matter that falls into the creek. And then as you float further and further downstream, you basically all of the stream starts to get more and more murky. So there's actually less and less algae being formed. And what you have at the very lower reaches of the stream are lots and lots of fine particulate matter that kind of just float in the water. And so you'll find more and more collectors out there than you would grazers and shredders. And if you notice that all the different reaches of the creek, there are different species that serve as either collectors or grazers and things like that, because each section of the creek, when you think of it on a very, very wide regional scale, there's different types of water quality that, that these organisms are suited for. And not only do these macroinvertebrates cycle nutrients through the aquatic food web, they also serve as a crucial food source for terrestrial ecosystems as well. So a lot of these macroinvertebrates spend a lot of their lives in the water as larvae. However, when they want to become adults, they will grow wings and they will fly out of the water and into the terrestrial ecosystem. And when they're doing that, they are fresh food for spiders and bats and birds and lizards. And so not only are they a good food source for the fish that live in these aquatic ecosystems, but when they become adults, they also serve as food and nutrients for other animals as well. The macroinvertebrates that we know of can also tell us a lot about the health of the stream. And what I mean by health, I mean by related to human impacts and how humans have altered the landscape. This could be urbanization and our non-point source pollution that ends up in our waterways due to our lifestyles. It could also be changing land, developing land for agriculture and allowing grazing next to a creek and things like that. And so scientists have determined that there are certain macroinvertebrates that are very sensitive to these pollutants. Pollutants could be fertilizers, it could be even like alterations in water temperature, like if there's like a nuclear plant nearby and they're using water to generate electricity or to cool their generators, and discharging all of that warm water into a river, that can also impact the water quality if it's not natural there. Um, so there are organisms that are sensitive to certain pollutions and alterations to the creek, and we call those pollution sensitive individuals. We also have individuals, macroinvertebrates, that are found to be pollution tolerant, so they can tolerate a lot of pollution and alterations to the creeks, and they will still be able to survive. And then we have invertebrates that are kind of so-so in the middle. And so they can tolerate a little bit of pollution, but not constant. So you might find those. So when you go to a creek, you can actually sample these macroinvertebrates and see who is around. If you find a lot of invertebrates on the bottom row, that might give you a hint that maybe this waterway is polluted and not as healthy. But if you find organisms in the top row that are super pollution sensitive, then you might be able to say that, hey, maybe this creek actually is okay. Um, Basically, a healthy creek supports a lot of biodiversity. So if we were to go to a creek, if we were to go to a really healthy creek, we would actually find all of these organisms and not just a few. Um, Scientists also, when they collect these macroinvertebrates, when they want to learn more about the health of the creek, we actually calculate something what is called a biotic index. So every single macroinvertebrate, it could be very fine, like every single species is given a score, a pollution tolerance score from 0 to, I want to say 10. Um, and then basically, um, how many of the same species you find, you will then multiply it by their pollution tolerance score and then divide it by how many of those species that you found. And then if you sum them all together, you actually get a score between 0 and 10, and that is what the HBI value is. And it's called the Hilsenhoff Biotic Index, if anyone wants to look more closely into that. And basically, that score at the end, you read the table of and compare the number that you calculated and what the table says. And so you can then determine if there might be some degree of organic pollution in your creek, depending on what species you find in that area. Deb, please let
Oh,
me know if there is.
sure. We don't have any questions from people in chat, but if anyone does have a question for Shelley, you're welcome to unmute yourself and go ahead and or raise your hand and I'll unmute you. But I do have a question actually, Shelley. So you
Okay.
just talked about this HBI value and that 10 is really poor quality and 0 is really low. My question is, can you give some examples of creeks around here that have a very low, like less than 5 HBI in the South Bay or the peninsula where you've been working?
Oh, great question. I actually have not gotten my toes wet and sampled a lot of the South Bay creeks. I do a lot of this scientific investigation. I used to do it a lot in Redwood City. We would have high schoolers who were either taking biology or AP environmental science to come out to their local creek and do this exercise with us. So we would collect the macroinvertebrates, identify them, and calculate the HBI index. And it was already like pretty mid-level. It was not uh, a perfectly healthy creek, but it wasn't polluted either. I would say another watershed that I'm in most of the time is Stevens Creek, and that creek has a reservoir that is basically altering the water quality downstream of where that reservoir is. And you can definitely see a difference of macroinvertebrate species above the reservoir versus in the lower reaches below the reservoir.
Well, that's really interesting to learn about with regards to how the reservoirs can really impact water quality just by being a barrier. So Alex has a question. Go ahead, Alex.
Yes, relating to the pollution levels. how do we tell if a certain invertebrate is in one of those levels without like looking at the water?
Without looking at the water. So, there are these giant species lists that different either nonprofits or agencies have published because there's different macroinvertebrates, different species that live in different parts of the world. So California specifically has one list that has all of the California species, and they've determined which macroinvertebrate is more pollution sensitive to others than others. All right, part two. So let's learn some macroinvertebrates. So as Deb alluded to, I spent a lot of time doing macroinvertebrate photography. I basically was 1 year graduated from college and I got hired by the lab that I still work at UC Berkeley. They were undertaking a really big study to try to determine the aquatic food web of this huge watershed called the Eel River in Mendocino and Humboldt County in Northern California. And basically, I was paired with a lot of other grad students and other ecologists who were there to help identify basically everything that was in that river. And they needed to know, like, what are the macroinvertebrates that live there? So I spent my summer in the South Fork Eel River in a little preserve called the Angelo Coast Range Reserve. It has some really beautiful creeks and a river there. And I basically walked out to the creek, walked out to the river every single day with my camera, and then I just looked for macroinvertebrates in the stream. And if they were nice, like I got to take really good shots of them, and I got really good at finding them. Um, I had a macro lens on me, and so yeah, it was just a really cool experience. I got to know a lot by identifying them all. I never had to preserve them because I could just use my macro lens and take specific parts of each macroinvertebrate from where I can then just use a dichotomous key and see like what species were they. This guide, I will share a little excerpts out of it, definitely not the whole thing tonight, but if you want to download the whole thing and when you're out by a creek and you want to know some other individuals that might be out there, you can download it at the website that I have below. And it will take you to a web page and you just scroll to the very, very bottom, and then there will be a little download link there. But tonight I'm just going to share a few pages from this guide, and then I will talk about the different groups of macroinvertebrates based on their like taxonomic classification. So the first group of macroinverts I want to show you are the Ephemeroptera. So this is an order of insects. Their common name are the mayflies, and they are super, super common. These are pretty much the most abundant group of organisms that you might find in a stream. There are many, many different families and different species that inhabit the creek, but they're pretty much found in all different habitats of the creek. I want to share this family in particular, the ephemerellids. If you can allude to their name, ephemerellid is named after the word ephemeral. Ephemeral means something that's super temporary, and is pretty much gone in an instant. So a cool fun fact about these invertebrates is that this family in particular, they are a univoltine, they exhibit a univoltine lifestyle, which means that they basically have a whole generation from eggs to larvae to adult, all in the course of 1 year. And specifically the ephemerellids, you pretty much can only find them in a creek in the springtime. After the springtime, all of them have pretty much um, died off, and they're now just incubating as eggs in the bottom of the creek. So this is why they're called the ephemerellids, because if you're out in a creek, you'll only see these in the springtime. So this is a mayfly. a burrowing mayfly. We probably won't see these species in California. This is from the BBC, but it shows how they transform into adults. They basically kind of float to the top of the water, where then they shed their their larval exoskeleton, and they kind of grow wings and transform like this. They'll probably spend some time at the surface of the water to kind of dry off their wings, but right after that, they're ready to fly. And then what is really cool is that a lot of mayflies, they do what is called swarming. So a bunch of the same species usually will get together. Usually we'll find this in the early morning and the evening, or the evening by a creek. And you will just find these like swarms of mayflies. You can see how some of these mayflies are just flying up, and then they're floating down and flying up and floating down. And this is the male mayflies actually looking for a mate. They mate on the wing. They'll find a female mayfly, and they will mate in the air. And then after they're done mating, the females will actually go back to the water to deposit their eggs. And usually their adult life usually spans over the course of 1 or 2 days. And after that, they're pretty much done. They become fish food, and they don't really live long past after 1 or 2 days. The next group of macroinvertebrates I want to show you are the odonates. So these are the dragonflies and damselflies. These are really ferocious predators in the aquatic ecosystem. They're always looking for other smaller prey items to eat. To tell the difference between a larval dragonfly versus a larval damselfly is that damselflies will have 3 pairs of gills at the end of their bodies. So the 2 photos on the lower row are examples of damselflies. And then dragonflies, they won't have that. Instead, they'll just have a ring of little spikelets at the bottom of their bodies. And they use that. They can actually siphon water and propel themselves through the water by jetting out a stream of water through their butts like that. And then I also want to show you all how these dragonflies and damselflies look for prey. So here we have a dragonfly. Some of them like to bury themselves in the sand to camouflage themselves, but they basically have this really interesting mouth part that they use to capture their prey. It's kind of like an arm extension almost, where they can basically propel their jaws forward and capture prey and bring it back to their mouths. So we can see one dragonfly here using their eyes to look for prey. And then almost instantly it grabs something out of the water. This action is super, super fast. So you're looking at a slow-mo video of this. But they're really, really accurate predators, and they rarely miss. And sometimes they can also take down other bigger things like tadpoles. So we think insects getting eaten by frogs, but sometimes the insect is the one eating the frog. Next, we have the stoneflies. These are also a group of really beautiful macroinvertebrates. Like the mayflies, they spend a lot of their aquatic young lives in the water, and then they will also come out of the water and transform into adults and do kind of the same life cycle. So there's many, many different species of stoneflies that live in creeks. And that is the most I can talk about them. Stoneflies might look very similar to mayflies, but once you get a sense of how they look like, you'll be able to differentiate the two. Stoneflies usually will only have 2 tails, And they will also only have 2 claws at the end of each leg, compared to mayflies that will probably have, most of the time, will have 3 tails, sometimes 2. And then they can also get pretty big as well. We have two pretty common stoneflies in California. We have the pearlids, which are the common golden stoneflies. And then we have the giant stoneflies called the salmon flies. And these two families, these invertebrates, actually live underwater for close to 3 years before they decide to turn to adults. So that is why these stoneflies are pollution sensitive bioindicators, because of that. Since they live so long in a creek system, if there's like any sort of pollution, we'll basically lose a whole generation of them, and so we won't be able to see them. Next, I'll talk about true bugs. We have true bugs that live in our creeks. Some of them are truly aquatic species, meaning that they live in the water. And then there's also some that live on the water. So if you've ever been to a creek before and you've noticed that there's some insects that actually look like they're walking on water, this could be one of them. It's called the water strider. Water striders are really interesting true bugs because they have evolved to basically float and walk on water. They use their middle legs to kind of propel themselves like oars on a boat across the water. Also, their legs have a lot of fine hairs on them, and so that is how they're able to not sink in the bottom of the water. Water to an insect is actually really, really sticky. So a lot of insects get stuck when they touch water. And these water striders are actually here to eat all of the insects that are stuck on the surface of the water. So they have a long piercing mouth part. They'll basically just pierce their mouth parts into insect, and they will suck all the juices out until there's no more. The next group of macroinvertebrates are the megalopterans, the hellgravette stops and flies in all their flies. These are quite rare species found in creeks. A lot of them really need cold water to live, and so you'll find them under rocks in very, very cold environments underwater, but very rarely do you actually find many of them. They can get really big, and you can tell them apart from other invertebrates by what are called these lateral filaments. These lateral filaments, pretty sure we still don't really know what they are or what they actually do, but they look really cool on these invertebrates. Next, we have beetles. There are a lot of beetles that live on land, but there's also many beetles that live in the water as well. There's some beetles that look really, really flat. These are the water pennies. So if you ever pick up a stone in the creek and you find there's like something flat that's moving around, that's probably a water penny. You'll also find different beetles at different stages of life. The two beetles that are on the right-hand most side look almost worm-like, and so those are the larval stages of a riffle beetle that will actually metamorphose and turn into what we typically know look like, what a typical beetle will look like. Some beetles don't ever leave the water after they become adults. Some of them actually stay in the water all their lives. This is a predaceous diving beetle. It doesn't have any gills per se to do gas exchange. Instead, it keeps a little air bubble tucked behind its wings so that it can breathe underwater. This is a long-toed water beetle. Instead of tucking a little air bubble behind its wings, it actually has a lot of fine hairs that actually trap air and envelop the entire insect in like a little spacesuit of air so that it can continue to do whatever it wants to do underwater and still be able to breathe. And so that's why you'll find this like shimmery golden look to it. Next, I want to talk about true flies. So these are in the order Diptera. A lot of the true flies, when they're larvae, they kind of look like worms. And they're also very, very common in aquatic ecosystems, specifically the midges. So the midges look like worms, except that they have a small encapsulated head at the very top. So you can see that they actually have a head. They don't actually have true legs. Instead, they just have some appendage that is called a proleg. And then you can see it usually swims by thrashing its entire body in the water so that it can move. There's some midges that have evolved to become filter feeders. The Rheotanitarsus, the midges on the right, they actually make a house around them, a small little tube, and at the very end of their tube, they have these really, really long spindly attachments where then they spin this little web of sorts that catch organic material or other floating things in the water that they will then just pick off their little net and eat. There's also some midges that have a symbiotic relationship with certain cyanobacteria, specifically the Cricotibus species that lives on nostoc. Nostoc is a type of cyanobacteria that's really common in California streams. When you go to a stream, usually you'll see a rock and you will see these like little blue-green ear-like things attached to the rock. And if you look really, really closely, you might be able to find a midge that is living inside the cyanobacteria colonies. On the right-hand side, we have Pseudochironomus richardsonii, which is also another type of midge that actually weaves a home out of Cladophora type of algae. And that's how it can protect itself against predators by encapsulating itself in this algae. And then we also have midges that actually parasitize other macroinvertebrates. The Symbiocladius midge actually burrows itself into a mayfly, the Ecdionurus species of the flat-headed mayflies. And they basically stay there. They just look like a backpack. And it just feeds off of the hemolymph of this mayfly. It never kills it outright. However, as it's taking all the energy away from this mayfly actually won't have enough energy to transform fully into an adult. We also have different types of true flies that live in the water. Mosquitoes actually are very rare to find in river ecosystems because they require very still water surfaces. When I sample creeks, I very rarely come across mosquitoes, but if you have like a stagnant pool somewhere, or if you're sampling really close to the banks where the water doesn't move as much, you might be able to find them there. But rarely, if you're in a moving creek, you won't find mosquito larvae. However, you might find a lot of blackfly larvae. Blackfly larvae really like to situate themselves on the fastest flowing parts of creeks and rivers. You'll find them where the current is really fast. They have this adaptation where they will basically secrete this glue-like adhesive out of their mouths and plant their abdomens or the very bottom of their abdomens onto the rock. And so that's how they're able to attach themselves to things without getting swept by the current. After the true flies, I'd like to talk about caddisflies. These are basically the architects of a creek ecosystem. A lot of them are caterpillar-like in shape. their larval form will look very closely resemble like a caterpillar because they're very closely related to butterflies, actually. Many of them, depending on the species, will build cases or what we call retreats around their bodies to protect the most vulnerable soft parts from predators like turtles and fish. And depending on the species, they will actually hone in on different materials to use. So there's some that will only use small little pebbles, and they will arrange them neatly so that their case is very smooth. Some will basically use twigs. They'll cut up little leaves and arrange it around themselves. And then there's ones that are just like, I'll put whatever's around and then I'll make a case that way too. There's more caddisflies that actually do not make a case around themselves. These are what we call naked caddisflies for obvious reasons. But instead of making a case that they drag around them throughout their lives, they'll actually make a shelter on a rock, and they will live in that shelter throughout their lives. Some of them will actually move. They're quite mobile, and so they'll be free crawling, and then they will just move around and find whatever they're to eat. What's really interesting about caddisflies is that when I was talking about how caddisflies make cases around themselves, they actually secrete a special protein to glue all of these pebbles together. There's chemical scientists are really interested in this type of adhesive because they think we can find beneficial uses to it. To find like a glue that is both waterproof and very strong, it can actually be used in underwater situations. When we think about surgery and having to attach like tissues back together instead of staples, we can try to borrow what caddisflies have or synthesize the proteins that they're using so that we can advance surgery and other medicines that way. Next we have the butterflies and moths. These are insects that we typically think that are flying in the air, and most of them are. However, there's a few species that actually spend their young years in underwater in creeks. You won't find them free floating or free crawling. Instead, they make what is a silk retreat on rocks where they will spend their entire lives living inside and feeding on whatever is growing on the rock. To me, their retreats kind of look like snot. So because it's just like very translucent and you might see something wriggling inside. When they begin to pupate, they will transform their silk retreat into a cocoon, which is more harder and more clear to see. But after they're done metamorphosizing and turning to adult butterfly, they kind of look like this. And you can actually find them flying at night if you ever put out a black light at night to attract moths. You'll be able to find some if you're doing this nearby a stream. And then my following few slides are all invertebrates that are not insects. So we have a bunch of organisms from the phylum Mollusca. They are the snails, the limpets, and the mussels that live in the creek. They come in all different shapes. We have the segmented worms, the oligochaetes. We have ones that look like regular earthworms. We also have unsegmented worms that are mostly parasitic, like the horsehair worms and nematodes. And then we have flatworms that live in the creek as well. Another segmented worm is the leech. We do have leeches in California. They are actually very small. They do not feed on human blood. Instead, they predate on snails. So they will hunt down snails and basically eat their insides as they are swimming throughout the creek. We also have things like crayfish and amphipods. So these are scuds. So we're kind of like diving into the planktonic part of the creek, who lives in the creek. And we have water mites, and we also have seed shrimp. And we also surprisingly have freshwater sponges as well. My final slide is basically to highlight all of the different other things you might find in the creek. So a lot of these invertebrates lay eggs, and I've spent a little bit of time studying about who lays what type of eggs. And you can see just all the different variations and shapes and colors that you might see when you turn over a rock in the creek. So that is the end of my part 2. I know that wasn't all of the invertebrates you might find, but it was basically a speed run of all the major groups that you might find. So if anyone has any questions about that, let me know.
Yeah, thanks so much, Shelley. That was fascinating to see your amazing photography as well. How big are the sponges?
The sponges that I've seen are pretty much, I've never seen them larger than maybe the size of a coconut. We're thinking about the size of different fruits. They're usually small. They don't grow into like big columnar ones like the marine sponges do. They're kind of more like mound forming, and they're very amorphous in their growth form. They're also very, very rare. Their habitat has definitely been destroyed or degraded by humans, and I've only seen them in like very, very pristine creeks.
So you talked about a dichotomous guide that you used. Is there a name to that guide that you can share with us?
Yeah, it's basically the textbook of all aquatic macroinvertebrates found in North America. It's colloquially known as Merritt and Cummins. Those are basically the two main authors. And I believe the title is, we could get this wrong, An Introduction to North American Aquatic Macroinvertebrates.
It's another question. Do you need a permit to search creeks for macroinvertebrates with students?
Ah, that is a great question. Yeah, it's really interesting on just how water is managed throughout California. In Santa Clara specifically, that I know of, in Santa Clara County, the local water agency, Valley Water, they'll always require a permit of some sort, even just to like enter the creek. But in other counties, where there is no like water agency, you're pretty much free to just like go into a creek in a park and flip over some rocks and see what you find. As long as you are not transporting these macroinvertebrates to like a school or another area, if you're just looking at them and then immediately releasing them, keeping them alive and putting them back where you found them, then you don't need a permit. If you're specifically looking to like preserve these macroinvertebrates, either for like a museum collection or to study them further, you'll definitely need a permit from the California Fish and Wildlife, because they are always tracking who is taking things from these natural ecosystems.
It's pretty amazing to need a permit for something so small, but I guess everything counts, right? Every little bit.
Yeah.
All right, the next question that we have is, do any of these macroinvertebrates tolerate some level of salinity, like at the lower reaches that feed into the bay?
Yeah, so that I can highlight some of my research that I got to help out in during 2020, 2021. We were actually researching the macroinvertebrates that live in the Eel River estuary. So the estuary is an area of the river where seawater from the ocean mixes in with the fresh water of the river. And you basically just get a whole different assemblage of life there. A lot of the insects, I believe, that are macroinvertebrates cannot survive that type of brackish water. Instead, you'll get different macroinvertebrates like more scuds. You'll get more marine life like oligochaete worms, polygochaete worms, a lot of isopods as well. So those are like aquatic roly-polies that live out in the estuary. So definitely more marine life will live in the estuary than what I presented today. That'll probably be a Shelley Guide Part 2. I need to go out and document all of those.
Another thing that came up for me as you were talking is how long have these types of critters been around? 100,000 years, 1 million years, a billion years? What's the life scale of these orders of beings?
In terms of evolutionary history?
Yes, yes.
Okay. So let's go back to this this phylogenetic tree right here. When we think about evolution, we're thinking about millions and millions of years. And so this is a tree that basically tracks when certain types of organisms evolved. So if you kind of look at the 3 o'clock position of this tree, you basically get the organisms that are close relatives of insects. They're actually called hexapods, like the Dipleura, the Protura, the springtails, and the bristletails. And then as you go in the clockwise direction, you'll see that mayflies, dragonflies, and damselflies come next. And this tree is arranged that the more earlier the organisms, the insects that evolved earlier in evolutionary time, start off where that is. So scientists believe that aquatic invertebrates or insect life in general started in fresh water. The mayflies and the dragonflies and the damselflies evolved first. And then when time went on, a lot of insects ended up becoming terrestrial. Like we had the formation of plants, and then the plants evolved flowers, and then we had a giant radiation of evolution there with pollinators. And so the butterflies and the moths and the caddisflies, they actually, we believe, their ancestors started life on land. However, they came back to the fresh water. They evolved to come back to and return to their lives, or evolve a different species to be aquatic. Does that make sense?
And what was the time period of them going from 3.4 billion years ago, there being the beginning of life as single cell organisms, to when their cells came together as at the 3 o'clock location?
Yeah, I don't quite remember this paper, but my best guess, if you look at the center of this tree, you can see that there are like concentric rings, and each concentric ring, I believe, is 1 million year. It's either like 1 million year or 10 million years. So if you go back to the center, it's basically very far back in time.
I just found it really interesting when you're talking about the caddisflies and how humans keep coming back to nature to look for our own solutions to some of our problems. And are there other examples of the macroinvertebrates that you shared that might fit in that category of where people are going back and looking at them to see if they have a property that we might want to exploit?
Materials engineers are always looking for new things. So not only are they looking at caddisflies and their very sticky silk, but they're also looking at clams and how they anchor themselves into the water. I also want to say that certain filter feeders that are macroinvertebrates, scientists are also looking into to see how we can better clean our own water for drinking using their techniques as well. Yeah, thank you for all these good questions. So my next and last section that I'll talk about is iNaturalist and how we can get these macroinvertebrates to be more represented on iNaturalist. A few days ago, I pulled this screenshot off of iNaturalist, basically detailing what are the observations of stoneflies in California. And if you look at the graph, not much. There's like less than 100 verified observations, and total observations overall is less than around 700 or less than 1,000. So, and I know that there are more people out there who are really who are naturalists and who want to take more photos, and hopefully like just giving the knowledge and where to look, and maybe hopefully giving you good photography tips on how we can better observe them with our cameras, and hopefully people will be able to identify them better, but it's always a challenge. So this was kind of the equipment that I was bringing out to the creek every day as I was taking aquatic macroinvertebrate photography. I was working in a pretty remote place, so everything had to fit in my backpack. I really couldn't keep um, big things. I was using a really old camera, I believe a Canon EOS 40D. It surprisingly only had like, I want to say 8 megapixels on it in resolution, but they were really good megapixels. So I highly suggest if you're ever getting into like the more amateur field of photography, just think about getting a non-smartphone camera and start playing around with it, because you'll definitely get better photos. You basically also want to take these photographs with the macro lens, because a lot of our invertebrates are small, and we're always looking for different small features on its body, either observing the hairs on its leg, and a macro lens definitely helps with getting that magnification. I was also carrying around aquarium net. You can easily find this in a pet store. This is used to like transport fish around from fish tank to fish tank. Find a net that has like a very fine mesh fabric so that you can capture more smaller things instead of them getting through the holes and you missing them. Um, I had several tools to manipulate these macroinvertebrates with. I had a flimsy forceps. These are just forceps that are made out of really thin sheet metal. And basically when you carry stuff with them, like they will not crush anything. They'll basically just gently envelop whatever you're trying to hold and won't crush anything. And so it's good to manipulate macroinvertebrates because some of them can be very soft and fragile. I was also using a pipette. I cut off the tip of the pipette so the aperture could be a little bit more wider. But for the really small macroinvertebrates, you can kind of like use the pipette to vacuum them up from the water and then place them into something that you're going to hold them for as you photograph them. You can bring along like a tackle box that you can sort out your photography subjects. So I had some that I placed in there and I was waiting, they were waiting their turn so that I can photograph them. And then my studio, as you would for these macroinvertebrates, is basically a cereal bowl. You just get one from the kitchen. I had a white background on it because it provided the most contrast. I had a ruler as well that I could lay at the bottom so I can get a sense of scale as I was photographing. Most of these ruler shots were edited out in my photographs, but you could see some of my photographs had like a little scale bar, so you can actually see exactly how big each macroinvertebrate was. When taking macro shots, so lighting is very, very important because as you're zooming in on a very, very small subject, you lose a lot of light, and a lot of your photos without extra lighting look very, very dark. So I didn't really have any special ring lights or anything. Instead, I had my headlamp that I would take camping with me. And so I just turn it on, and then I would just hold it right next to the camera lens, and it would shine light on my subject to illuminate it so that it would be much more brighter. And yeah, that's pretty much my setup that it was. And so this was like way back in the 2018, 2019. And so nowadays, I learned that on Amazon, you can get what is called a digital microscope. And macro lenses, when you get professional ones, it can cost like $300, maybe a little bit cheaper if you bought it used. But on Amazon, these digital microscopes can pretty much give you almost the same resolution. Might still be a little, there might be some aberrations here and there in your photo quality, but they're only like $40 and you can get pretty big magnification. It hooks up to your phone so that you can bring it out to the field with you and you can snap photos on your phone through this digital microscope. And then this digital microscope also runs on batteries, I believe. So you can recharge it and then take it out with you. And so it doesn't need to be hooked up to any like electrical source as well. When you're out in a stream, where do you want to look for these macroinvertebrates? You can look under rocks. If you see a submerged rock in the creek, you can take it out and take a look to see what is walking on that. Basically, just to reiterate, when I want to photograph them, I actually want to photograph all my subjects when they're still in water. That's the best way that you can get their natural appearance. And you also won't get all of that harsh reflection of water when they're above the surface, when they're wet. But if you have them under the water, you'll be able to get a better photograph that way. So usually my bowls or my sorter trays, they'll always have like a little bit of water on them, in them, so that my subjects can stay in their aquatic environment and wait patiently for me to photograph them. So yeah, so looking at rocks, taking rocks out of the water is a good way to look for macroinvertebrates. There's certain creeks have areas where maybe there's like a lot of leaf matter that accumulated in a certain spot. It might have snagged against the rock, but there's just leaves accumulating there. So I encourage you to like take your hands, grab a chunk of leaves and put them in your bowl or put them in your tray and see who is eating those leaves. In areas where it's sandy or a little bit muddy, you can actually take an aquarium net and basically hold it so it's perpendicular to the flow of water and also perpendicular to the ground. And then right in front of the opening of the net, you can then disturb the substrate. You can use your hands or you can use your feet to kick at the bottom and disturbing all of that sediment and all of that stuff, and even smaller things will rise up and get trapped into your net. Then you can empty that net into your tray full of water and you can see who is out there. And it kind of looks like this. We take high schoolers out pretty regularly to look at their local creeks. And so here is them. We just sampled a small creek in Palo Alto. The sample kind of looks like this, just lots of fallen leaves, some twigs. But there's also small things that wriggle around. And so they're really fascinating at see what is in there. And for example, this is what we found in one of the trays, a really, really small caddisfly. And this was taken with like just a regular cell phone. And we have pretty good resolution nowadays that if you just zoomed in and cropped a little bit, we can see a little bit more detail. That is the end of my presentation. Hopefully we will be able to get more observations on iNaturalist with the knowledge that I just shared.
Yeah, thanks, Shelley. All right, so feel free to turn on your camera, ask questions, say hi. It's always nice to see people's faces.
Hi, Shelley. I loved reading about on your website how on Eel River, it was hypothesized that some of the creek was classified into spawning or like fish spawning and then post spawning and flood. Do you think that that might be something that could be applicable, say, in San Francisco Creek?
Oh, great question. Are you specifically talking about the study of the caddisflies?
Yeah, in the link that you put for Shelley's book.
Mm-hmm.
On the top of it, it talked about the classifying Eel River in these stages.
Yeah. Oh, we study the Eel River on like a very, very large scale, like starting from its really, really small creek tributaries and all the way going down to the main stem. San Francisco Creek, I feel, is much smaller in scale, like a quarter of what the Eel River is.
Yeah.
But I am pretty sure we can. There is San Francisco Creek does have a dam, right? The Searsville Dam. So that might also like alter whatever results we might find because it is like a human impact that might yeah, we might change whatever we hypothesize. But I believe it can be done.
Okay. I think it might be cool. Just, I know Stanford's doing some studies on trying to see if salmon are in the creek. We haven't seen them for a couple of years now. We know they were there, but if we see indicators in the macroinvertebrates, that might be lines of evidence that the creek is still salmon or steelhead.
So Gary, would you like to add anything? One of the things when you come out to our BioBlitz events is that you bring a net and you catch some critters and throw them under a microscope.
Yeah, and the digital microscopes have come a long way in the last 2 or 3 years. They now have some that have multiple lenses and even a lighted stage, so you can get down and see the zooplankton, the rotifers, and other critters running around them. And they're 28 megapixel cameras, so they do a superb job. Those go about $150. Some of them do have screens, and the one I take out to the BioBlitz has a 7-inch screen on it, so you can look at stuff live. I also did it with trout in the classroom. Kids loved it.
Yeah, it's actually Gary, the one who I saw him and his setup, and I got really jealous. And he was like, yeah, it's really affordable. And it basically took better photos and better resolution than what I was having.
And like I said, they've come a long way in the last couple of years. They've gotten even better.
Yeah.
I don't have any questions, but I wanted to say thanks so much for the presentation. I learned a lot, and I'm going to tell all my friends now that dragonfly larvae have arms, weird arms attached to the front of their faces that they eat things with. Yeah, those videos were so cool, and I'm excited to check out the guide. And try to learn a little bit more about the macroinvertebrates.
Yeah, thanks, Ren. A lot of my videos actually come from KQED's Deep Look. KQED is our like local public radio broadcasting organization, but Deep Look is one of their YouTube channels where they basically do these really cute little documentary shorts of the really, really small things that we might overlook every single day. And so they've done a lot of small documentaries on aquatic insects, aquatic macroinvertebrates. They also do things like bees and other things as well. So highly recommend to go and watch some of their videos. They're really short, really sweet, and really great cinematography. What I shared today was like very, very like basic, very textbook information. A lot of like the new research that I'm really interested in that other people are researching nowadays is like what happens when the stream dries up? Like when there's no more water, where do the macroinvertebrates go? They all don't die out. They actually like burrow themselves deep like into what we call the hyphoreic zone. That's the area where that is under the surface, but there's still groundwater moving that we can't see from above. And so there's just more and more studies. We have better technology that we can sample down into the cobble layer, into that zone to see what a macroinvertebrates hide out there. And what do they do like when our summers dry up our creeks? So that's like another really interesting thing that we're just learning about when it comes to aquatic ecology.
That's interesting that you bring that up about the creeks drying out, because historically a lot of them would dry out over the summer. And now, because we have dams that control water, they're perennially wet. And how has that changed the insects that and macroinvertebrates that would be in a creek naturally?
Yeah, I would say that definitely when you have changed the creek hydrology, where now there's perennial flow, you'll definitely see different macroinvertebrates. I'm unsure if you might increase diversity or lower diversity, but for example, I've done a lot of monitoring on Stevens Creek in the South Bay. They have a reservoir. Unfortunately, that dam does provide somewhat of a perennial flow to some stretches of the creek. However, the water that comes out of the dam is actually not suitable for a lot of macroinvertebrates. Number one, there's a lot of sediment that is trapped behind the wall of this dam, and the water that comes out of it brings out all of that fine sediment, basically making the water super, super turbid and not clear at all. So if you ever go to like Stevens Creek in the Cupertino area, Sunnyvale area, the water there is actually very murky, and that is because of the effects of the reservoir. And a lot of macroinvertebrates don't like that type of water. They won't be able to survive. What we get are macroinvertebrates that are more filter feeders, so they can actually filter off food that come through that type of murky water. That murky water also makes it hard for algae to photosynthesize, so we get less and less of those macroinvertebrates who depend on algae that they can graze on. The water that comes out of reservoirs is usually also very much warmer in general, because the water stays behind that wall and it bakes in the sun. And so the water that comes out and continues down the creek is also warmer as well, artificially warm. And so that also like alters what can live in the creek, because usually you would expect creek water to be cooler than what is coming out of the reservoir. And then like another thing as well, this is another thing, the reservoir at Stevens Creek also has their output pipe at the very bottom. And so the water at the very bottom of the reservoir is actually anoxic. There's no oxygen in that water that is getting pumped out, which means that no oxygen and no organisms can survive except for the anaerobic bacteria. And these anaerobic bacteria, some of them are the methanogens. Those are the ones that basically transform inorganic mercury that's found in the natural bedrock in the mountains here. And they turn them into methyl mercury, which then becomes a problem in our food webs, where they bioaccumulate in the tissues of organisms, and they end up like poisoning birds and fish in the long run. So that's like my overall spiel about dams and reservoirs in general and how they affect biodiversity.
I have a quick question, just back to San Francisco Creek. there's two major branches of it, and only one of them is dammed. And I was just wondering whether anybody had looked at Bear Creek versus just upstream of the dam, and compared that to San Francisco Creek or to Corte Madera Creek below the dam. Just curious, because that's kind of a natural experiment.
I actually had the privilege of sampling at Bear Creek. I was with a group of Woodside High School students, and we got to get access to Bear Creek behind the elementary school at Woodside. And there was a lot of really great macroinvertebrates. We saw a lot of biodiversity, a lot of stoneflies and caddisflies, different species of mayflies. And then downstream on San Francisco Creek, like since only half of like the tributaries of San Francisco Creek are blocked by that dam, we actually I would say we don't really find like as severe impairment when we look at macroinvertebrates downstream. I sampled some macroinvertebrates this spring at San Francisco Creek, kind of where downtown Palo Alto is. We found a lot of pollution sensitive mayflies out there. We didn't find many caddisflies, especially the ones that shred leaves, but we found a lot of predatory caddisflies, so the ones that don't make any homes, but they like are around, crawling around and eating other organisms. And the water was also like very clear. Searsville Dam, they don't have a pipe at the bottom of the dam. Instead, it's kind of like a bathtub where where the water overflows down the walls. So we don't get that intense like turbidity that we see at Stevens Creek. And I think that has somehow like kept the aquatic ecology of lower San Francisco Creek a little bit stable.
So thank you everyone for joining us for the Naturalist Training, where hopefully you've learned a bit about all our wonderful critters that live in the creeks that are super small, the aquatic insects. And thank you to Shelley for sharing all of her knowledge, but also some of the tools and tips. And we're developing additional training events and plan to host more on topics such as insects and arthropods, which another entomologist, Marava, loves. And you can, if you have some ideas of things that you'd like us to talk about and get some presenters on, just drop me an email and I'd be happy to investigate. Part of the intent of these trainings is to have you not only learn more about what the topic is, but hopefully to come out and join us at some of our events and maybe be a docent.
Hi, that was really fantastic, really cool. I want to go again back to San Francisco Creek because of, the plans to open up the dam and recreate, water flow through that creek. I have access to an upstream branch of that, and I was just wondering, if people were looking at those upstream branches of the Corte Madera or Saucil Creek before they go into the Searsville Reservoir, and whether there's been any thought about laying a baseline. So laying a baseline before the dam gets opened. That's the question.
Yeah, I think I'm not in specific talks about about the Searsville Dam removal. And I know that there are scientists in that team who are trying to get like baseline information right now. And I really am hoping that they will be collecting aquatic invertebrate surveys out there. I know we've done a few water quality monitoring, community science water quality monitoring efforts out there in San Francisco Creek. And some people have wanted our data to kind of see like what are the current water temperatures, pHs, and things before the dam gets taken down.
Yeah, that's exactly the kind of thing that I was thinking. And I guess what I would like to know if there's somebody that I could talk to where I could, offer my location as either a research spot, or I could do the collection myself. That wouldn't be a problem, but it's quite a bit higher up. And it seems like it would be a good baseline to get. So.
Right. The person who I might connect you with who was in this webinar was Tess Byler. She works for the JPA, and I believe her organization is in the room when they discuss the Searsville Dam removal. I also
I think
know,
that's right. Yeah.
yeah, I also know that CalTrout is also one of the organizations that is consulting on the Searsville Dam removal. The local project manager for the Bay Area, I think his name is Ryan, might also get in touch with him as well, because they definitely would really appreciate that access to the creek, especially if it's like through private property.